Hello everybody, my name is Grace Pemble and today I will be presenting on pseudo-unipolar sensory neurons in the dorsal root ganglion and its development and regulation. I'll also be presenting on how the dorsal root ganglion can be a target for neurorestoration and neuropathic pain. Pseudo-unipolar sensory neurons have a variety of functions. One of these functions is in the role of sensory transmission. Pseudo-unipolar sensory neurons are specialized for transmitting sensory signals from the peripheral nervous system to the central nervous system, aka the CNS. These neurons relay information about pain, temperature, touch, and proprioception in the spinal cord and brainstem. Their structure allows rapid signal connection, minimizing synaptic delay because signals bypass the cell body during transmission. Another function is its signal conduction pathways. The neuron receives sensory input via peripheral axon terminals located in tissues, for example, skin, muscles, or joints. These signals travel along the peripheral axon, bypass the soma and the dorsal root ganglion, the DRG, and propagate through the central axon, entering the spinal cord via the dorsal root ganglion. When it comes to its structure, pseudo-unipolar neurons have a very unique morphology. Pseudo-unipolar neurons have a single process extending from the soma, which bifurcates into two branches. These branches are the peripheral axon, which extends to sensory receptors in the periphery, and the central axon, which projects into the spinal cord. This unique structure minimizes signal loss and facilitates high-speed transmission. It also has its cell body, which is located in the dorsal root ganglia, surrounded by satellite glia cells, which provide metabolic and structural support. Ion channels and receptors can also be found in this cell, expressing a variety of ion channels, including voltage-gated sodium, potassium, and calcium channels, which are crucial for generating and propagating action potential. These contain specialized receptors such as transit receptor potential (TRP) channels for detecting temperature and nociception. The dorsal root ganglion, aka the DRG, is located in the intervertebral forearm adjacent to the spinal cord. Each spinal nerve is associated with one DRG on either side of the vertebral column. Its projections, like the peripheral processes, innervate specific regions of the body, like the dermatomes, based on their location along the spinal column. Its central processes enter the spinal cord and terminate in specific regions of the dorsal horn to synapse with second-order sensory neurons. Now, when it comes to regeneration and injury response, after peripheral nerve injury, DRG neurons can regenerate their peripheral axons. But, on the other hand, central axons within the spinal cord exhibit more of a limited regeneration due to the inhibitory CNS environment. Molecular mechanisms like peripheral regeneration activate the growth of genes such as GAP43, which promotes axon elongation. Macrophages in Schwann cells clear debris and secrete neurotrophic factors like NGF and GDNF to support regrowth. Some challenges that may come up for sensory neurons can include central axon regrowth, which is limited by glia scarring in the presence of inhibitory molecules, for example, NOGO, MAG, CSPGS. Experimental strategies to overcome these barriers include neutralizing inhibitory molecules, enhancing intrinsic growth capacity, and modifying the extracellular matrix. Within the phase of regeneration and recovery, in the absence of full regeneration, DRG neurons may exhibit plasticity by reorganizing their connections or increasing receptor sensitivity to compensate for lost function. By addressing these distinct function, structure, location development, and regeneration potential, pseudo-unipolar sensory neurons in the DRG are critical for maintaining sensory input and serve as a model for studying neural repair. Thanks for listening. And if you'd like to learn more about pseudo-unipolar sensory neurons in the dorsal root ganglion and its development and regulation, many journals from the National Library of Medicine have peer-reviewed this subject exactly with the roles of astrocytes in the regulation of synaptic plasticity, memory formation, and neuropathic pain. Thanks for listening.